How are you doing today, students? I hope you are doing well. And please accept and prepare yourself for another day of learning opportunities. By the way, I am Mark Medino Tejome, your science teacher for today. In this video lesson, I'll go over another topic in genetics with you. Our lecture will begin with a short prayer.
Please bow your heads and let us pray. Dear Jesus, please show me how to spend this day sharing your love in every way. Help me to be kind to everyone, to play and laugh and have lots of fun, shining your light and giving your grace, sharing your joy with a smile on my face. Angel of God, my guardian dear, to whom God's love commits me here. Ever this day be at my side, to light and guard, to rule and guide. Amen.
And before we get into our new topic, let's have a look at the objectives of today's topic. By the end of the lesson, students will be able to define the term chromosome, identify the structure of a chromosome, identify the parts or functions of a chromosomes, recognize and name the physical features of a chromosomes, and recognize both normal and abnormal human karyotypes. And before we move on to the new topic, do you still remember our last topic? If yes, what is all about? Yes, Miss Soriano? Very good! Our last topic is all about gene mutation. We learned that gene mutation are changes in single DNA bases, or small intragenic deletion and rearrangements. These are considered to be heritable effects because they are usually not lethal to exposed cells. And we also learned that the classification of gene mutation is often used interchangeably with point mutations. Okay, since you are already known about our last topic, we can now proceed to our next topic. I prepared an activity for you to begin our new lesson. I'm going to show you some pictures and jumbled letters about our new topic. You've been given the duty of arranging those words based on the images. Each right answer has a time limit of 10 seconds. Is it true that everyone got the right answers to our first activity? Congratulations if you answered yes. If not, don't worry, I'll provide you with a variety of exercises to help you learn more about our today's lesson. Are you familiar with the word chromosomes? Or what comes to your mind when you hear chromosomes? By the way, my topic is all about the portrait of a chromosome. Chromosomes are thread-like structure located inside the nucleus of an animal and plant cells. Each chromosome is made of protein and single molecule of DNA. Passes from parents to offspring. DNA contains the specific instructions that make each type of living creature unique. Extra genetic material has milder effect on health that is deficit. Most large-scale chromosomal abnormalities present in a cell disrupt or halt prenatal development. Only 0.65% of all newborns have chromosomal abnormalities that produce symptoms. Cytogenetics is the subdiscipline within genetics that links chromosomes variation to specific traits, including illnesses. This chapter explores several chromosomes level abnormalities and their effects on health. Actual cases are used to describe some of them. Chromosomes have long been described and distinguished by size and shape, using stains and dyes to contrast dark and light region of the DNA. The essential parts of a chromosomes are telomeres, origin of replication sites where replication forks begin to form. The centromere recall from those telomeres are chromosome tips. Centromeres lie within vast stretches of heterochromatin. The arms of the chromosomes extend outward from the centromere. Gradually, the DNA includes more protein encoding sequences as a distance from the centromere increases. Gene density varies greatly among chromosomes. Chromosomes 21 is gene desert, harboring a million base switch with no protein encoding genes at all. Chromosome 22 contains 545 genes to chromosome 21st 225. Subtelomeres include some protein encoding genes and therefore reach the genes rich region, and the telomeres repeats. Area of 50 to 250 bases consists of 6 base repeats, each a few copies.
In humans, each cell normally contains 23 pairs of chromosomes for a total of 46. 22 of these pairs, called autosomes, look the same in both males and females. The 23rd pair, the sex chromosomes, the 22 autosomes are numbered by size. The other two chromosomes, X and Y, are the sex chromosomes. This picture of the human chromosomes lined up in pairs is called a karyotype. Karyotype is a preparation of the complete set of metaphase chromosome in the cells of a species or in an individual organism, sorted by length, centromere location, and other features, and for a test that detects this complement or counts the number of chromosomes.
The 24 human chromosome types are numbered from largest to smallest 1 to 22. 5 human chromosomes 13, 14, 15, 21, and 22 have blob-like ends called satellites. At least 500 proteins encoding genes lie in the total subtelomeres regions. Centromere possession distinguishes chromosomes. A telocentric chromosome has the centromere toward one end, although telomere DNA sequences are still at the tip. Humans do not have any telocentric chromosomes. An acrocentric chromosome has the centromere near an end. A submetacentric chromosome's centromere creates a long arm Q and a short arm P. And a metacentric chromosome's centromere establishes equal-sized arms. These multi-gene families may reflect present evolution. Apes and chimps have only 1 or 2 genes for many of the large gene families in humans. Such genes organization is one explanation for why our genome sequence is so very similar to that of our primate cousins, but we are clearly different animals. And before we proceed to our next discussion, we'll be having a short quiz for us to evaluate if you have learned something from the previous one.
Visualizing chromosome. Extra or missing chromosomes are detected by counting a number other than 46. Identifying chromosome rearrangements, such as an inverted sequence or an exchange of parts between two chromosomes, requires a way to distinguish among the chromosomes. A combination of stains and DNA probes applied to chromosomes allow this. A DNA probe is labeled piece of DNA that binds to its complementary base sequence on a particular chromosome. Obtaining cells for chromosome study. Any cell other a mature red blood cell which lacks a nucleus can be used to examine chromosomes, but some cells are easier to obtain and culture than others. Skin-like cells collected from the inside of the cheek are the easiest to obtain for a chromosome test. White blood cells are used too. A person might require a chromosome test if he or she has a family history of a chromosomal abnormality or seeks medical help because infertility.
Amniocentesis and chorionic villus sampling have been available for many years. A doctor removes a small sample of fetal cells with a needle passed through the woman's abdominal wall. Chromosomes microarray analysis proves and displays specific sequences detecting many disorders.
Amniocentesis. Amniocentesis is a procedure used to take out a small sample of the amniotic fluid for testing. This is the fluid that surrounds the fetus in a pregnant woman. Amniotic fluid is a clear, pale yellow fluid that protects the fetus from injury and protect against infections. 3 ways to check a fetus chromosomes. First, amniocentesis draws out of amniotic fluid. Fetal cells shed into the fluid are collected and their chromosome examined. Second, chorionic villus sampling removes cells that would otherwise develop into the placenta. Since these cells descended from the fertilized ovum, they should have the same chromosomal constitution as the fetus. The last one is, researchers can detect fetal cells, DNA, or mRNA in a sample of blood from a pregnant woman.
Down syndrome, also known as trisomy 21. Down syndrome is a genetic condition. It is not an illness or a disease. Down syndrome is caused by the occurrence of an extra chromosome, chromosome 21. This result is a range of physical features, health problems, developmental delay, and some level of intellectual disability. Screening for Down syndrome. Healthcare is nationalized in Denmark, and prenatal screening for trisomy 21 is available to all. In addition, all of the nation's 19 departments of obstetrics and gynecology use the same software of their record keeping, so that it is easy to maintain national databases of test results and clinical outcomes. Studies are ongoing to assess the societal and bioethical repercussion from implementing the new guidelines. Detecting fetal cells or nucleic acids in the pregnant woman's bloodstream is safer than amniocentesis and CVS, but is still experimental in the United States. Staining. Staining allows the proper visualization of chromosomes under these imaging techniques. It increases the contrast of the chromosomes. Moreover, staining can lead to banding, which is a consequence of differential staining along the length of the chromosome. Banding provides more information about the chromosomes. Fluorescein in situ hybridization, or FISH, is a laboratory technique for detecting and locating a specific DNA sequence on a chromosome. The technique relies on an exposing chromosome to a small DNA sequence called a proof that has a fluorescent molecule attached to it. The proof sequence binds to its corresponding sequence on the chromosome. FISH is more precise and targeted than conventional chromosome staining because it uses DNA probes that are complementary to specific DNA sequences. The probes are attached to molecules that fluorescent will illuminated, producing a flash of color precisely where the probe binds to a chromosome in a patient sample. Chromosomal short band. The shorthand way to refer to a normal set of chromosomes is 46XX for women and 46XY for men. 46XY, 46XX. At conception, an egg containing 23 chromosomes from the mother combines with a sperm containing 23 chromosomes from the father.
A human karyotype is abnormal if the number of chromosomes in a somatic cell is not 46, or if individual chromosomes have extra missing or rearranged genetic material. Abnormal chromosome account for at least 50% of spontaneous abortions, yet only 0.65% of newborns have abnormal chromosomes. Therefore, most embryos and fetus with abnormal chromosomes stop developing before birth. Polyploidy. Polyploidy is the condition in which a normally diploid cell or organism acquires one or more additional sets of chromosomes. In other words, the polyploid cell organism has 3 or more times the haploid chromosome number. Polyploidy arises as the result of the total non-disjunction of chromosome during mitosis and meiosis. The most drastic upset in chromosome number is an inter-extra set. A cell with extra sets of chromosome is polyploid. An individual whose cells have 3 copies of each chromosome is a triploid, designated 3N for 3 sets of chromosomes. 2/3 of all triploids results from fertilization of an oocyte by 2 sperms. The other cases arises from formation of a diploid gamete, such as when a normal haploid sperm fertilizes diploid oocyte. Triploids account for 17% of spontaneous abortions. Very rarely, an infant survives as long as a few days with defect in early organs. Aneuploidy. Aneuploidy is a chromosomal anomaly in which the number of 1 or more chromosomes is abnormal. Normal human somatic cells carry 46 chromosomes, 2 copies of each of the 22 autosomal chromosomes, and 2 sex chromosomes, either XX for female or XY for male. Monosomy and trisomy condition results from the subtraction or addition of chromosomal material respectively. Cells missing are single chromosome or having extra 1 or more aneuploid, which means not good set. Rarely, aneuploids can have more than 1 missing or extra chromosome, indicating defective meiosis in a parent. A normal chromosome number is euploid, which means good set. Extra missing chromosomes aneuploidy, unequal division of chromosomes pair can occur at either the first or second meiotic division. A single pair of chromosomes is unevenly partitioned into the 2 cells arising from meiosis 1 in a male. The result, 2 sperm cells have 2 copies of the chromosome and 2 sperm cells have no copies. When a sperm cell with 2 copies of the chromosome fertilizes a normal oocyte, the zygote is trisomic. When the sperm cell lacking the chromosome fertilizes a normal oocyte, the zygote is monosomic. This non-disjunction occurs at meiosis 2 because the 2 products of the first division are unaffected. 2 of the mature sperm are normal and 2 are aneuploid. Oocytes can undergo non-disjunction as well, leading to zygotes with extra or missing chromosomes with normal sperm cells fertilize them.
Following are the cases of the most common autosomal aneuploids among newborns. The most frequently seen extra autosomes in newborns are chromosomes 21, 18, and 13. Trisomy 21, or known as Down syndrome, results when cell division involving chromosome 21 occurs. These cell division abnormalities result in an extra partial or full chromosome 21. This extra genetic material, is responsible for characteristic features or developmental problem of Down syndrome. Another chromosome abnormality or another common autosomal aneuploids is trisomy 13, or known as Patau syndrome. Trisomy 13 is a rare genetic disorder caused by having an additional copy of chromosome 13 in some or all of the body's cell. Due to the presence of several life-threatening medical problems, many infants with trisomy 13 die within their first days or weeks of life. This chromosome abnormality appears to affect females more than males. most likely because male fetuses do not survive until birth. Another chromosome abnormality under autosomal aneuploids is trisomy 18, also called as Edwards syndrome. So Edwards syndrome is a chromosomal condition associated with abnormalities in many parts of the body.
Deletion and duplication. Deletion occurs when a chromosome breaks and some genetic material is lost. Deletion can be large or small and can occur anywhere along a chromosome. Duplication. Duplication occurs when a part of chromosome is abnormally copied. There are two major types of translocations, as well as rare types. In a Robertsonian translocation, the short arm of two different acrocentric chromosomes break, leaving sticky ends on the two long arms that join, forming a single, large chromosome with the two long arms. The tiny short arms are lost, but their DNA sequence are repeated elsewhere in the genome, so the loss does not cause symptoms. A chromosome, or a part of one, doubly represents one parent. It can result from non-disjunction in both gametes or from a trisomic cell that loses a chromosome, leaving two from the same parent. Uniparental disomy causes symptoms if it creates a homozygous recessive state associated with an illness or if it affects an imprinted genes.